Hello and welcome to all my dear students. I hope you all are watching the lectures and you have well understood the chapter Principles of Inheritance and Variation so far. Today, beta, we are going to study about sex determination, plus we are going to do the introduction part of mutation. Today's lecture is also going to be a small and crisp one, which is going to give you a very important information. So please stay connected throughout the lecture. Keep listening and keep making your own notes without a failure, which is very important, and I keep on informing you all about that simply listening to the lecture is not going to help you alone. Along with the lecture, you have to also make your own notes, plus practice the questions. You have to practice multiple choice questions and other types of subjective objective questions to just improve and enhance your knowledge. I hope you all are going to mark my words and will surely do the needful. Chalo, then let's start with today's class, which is actually lecture number 6 of the chapter Principles of Inheritance and Variation. Beta, today we are going to cover the sex determination. Introduction part I have already given you in my last lecture, that who tried to explain the concept of sex determination. It was done by Henking, right? So Henking was the person who introduced the concept of sex determination, in which he told, basically he was studying the spermatogenesis in some insects, where he found that 50% of the sperms were receiving the specific X body which he was tracing, and 50% of the sperm were not receiving these X bodies, right? Later on, these X bodies were termed as X chromosome. So this is basically the type of sex determination given by Henking on the basis of the nature of chromosome. So this type of sex determination is known as chromosomal sex determination. In some organisms like turtles and amphibians, I told you in my last class that sex determination can also be done environmentally. Basically, temperature plays an important role in deciding that whether the egg on hatching will develop into male or will develop into female. So like for example, in case of turtles and amphibians, sex determination is temperature based, not chromosomal based. It is temperature based, where the temperature, the range of temperature, whether high or low, is going to decide the nature of the sex chromosome, the nature of the sex of an organism. Correct? So all these things we have already discussed in our last class. Today, beta, we are going to start with proper chromosomal sex determination, which is observed in majority of the organism. That means the type of chromosome, genetical or chromosomal sex determination means the nature and type of chromosome is going to decide the sex of an organism, is going to decide the sex of an organism, right? So basically the type and nature of chromosome is going to decide the sex of an organism. Now it can be of two types, beta. One, male heterogamety, and second, female heterogamety. What is this? Male heterogamity means when the male is producing two types of chromosome on the basis of their sex chromosome. Male heterogamity beta means when males are basically producing two types of gametes depending upon sex chromosome. Depending upon sex chromosome. Whereas if I talk about female heterogamity means beta when female produces two types of gametes. When females produces two types of gametes on the basis of sex chromosome. Right? So there are two types of organisms, there are two types of sex determination, genetical sex determination, one under male heterogamity and under female heterogamity. Beta, under male heterogamity, we are going to study two types, XXXO type and XXXY type, where this XO and XY are representing the chromosome of the male, whereas XXXX, you can see these are the sex chromosome of the female. You can see the male chromosome are of different type. Here also this is the male sex chromosome and here also this is the male sex chromosome which is of different type. Hence it is classified under male heterogamity. Similarly, female heterogamity can again be of two type, ZZZO type and ZZZW type. So here the female heterogamity is this. So these are the sex chromosome of female. When female is going to produce two types of gametes on the basis of sex chromosome. So these are the four types that you have to study under male heterogamity and female heterogamity respectively. Chalo. So let's start first with XXXO type, which is under male heterogamity. Now beta, these are the sex chromosome written. Now, apart from a pair of sex chromosome, we also have certain pair of autosomes that are represented by A symbol. In a diploid organism, first of all, this is seen in case of grasshopper. Grasshoppers, beta, they show male heterogamity, that is XXXO type of sex chromosome, where male grasshopper in a diploid cell, they have this type of arrangement of chromosome where A is representing pair of autosome and XO is representing pair of different sex chromosome. Right? Now, when this diploid cell, suppose this is a gonad cell, if it will undergo reduction division beta, if it is going to undergo meiosis, then this male grasshopper is going to produce two types of gametes. It is going to produce two types of gamete. One gamete having X chromosome along with autosome and another having again autosome along with no sex chromosome. O means 0. 0 means no sex chromosome. X is the X sex chromosome. Right? So, grasshopper, male grasshopper beta, they show male heterogamety because their males are going to produce two types of gametes. One having sex chromosome, one without sex chromosome along with autosome. right? This is male heterogamety in grasshopper. Coming to the second type, which is seen in case of Drosophila, even we humans also show this type of male heterogamety. So human beings, Drosophila, shows male heterogamety, which is XXXY type. That means females are having, the diploid cell of female is having abnormal pair of autosome. Along with this, we have pair of sex chromosome where both the sex chromosome are same. That means if a diploid cell of such female will undergo reduction division, then every time they are going to produce same type of gamete having same type of autosome and sex chromosome, which is X. Correct? But in our males, what is the condition? In human male, in Drosophila male, the condition in a diploid cell is pair of autosome and pair of sex chromosome where both the sex chromosome beta are different. One is X, another is Y. Now, when such a cell is going to undergo reduction division, then it is going to produce two different types of gametes. One having X chromosome and another having Y chromosome. So this is known as male heterogamety. So one is about XXXO type of male heterogamety and second is about XXXY type of male heterogamety. These are the two different methods of sex determination on the basis of chromosomal arrangement, right? Next to set coming under female heterogamety is ZZZO type, which you see in case of butterflies and moths, right? And apart from this, second type of female heterogamety is ZZZW type, which we see in case of birds like hen, right? Now, first we are going to talk about ZZZO type. Here, beta, female heterogamety is existing, which means females are going to produce two different type of gametes, but males are going to produce same type of gamete. So the ZZ is the symbol for male and ZO is for female. Correct? Now what happens in case of female heterogamety? Female is going to have a diploid cell of female is going to have this arrangement of chromosome. Specific pair of autosome plus one pair of sex chromosome in which in pair only one sex chromosome is present, second sex chromosome is absent. Now beta, when such a diploid cell is going to undergo reduction division, what will happen? Two types of gametes will be produced. Can you name them? What will be the two types of gametes now produced over here? Any confusion? One gamete will have one of the sex chromosome along with autosome, and second type of gamete will only have autosome but no sex chromosome. So yes, this is known as ZZZO type. Now this is about females. Check about the males. In males beta, the arrangement is ZZ type. That means a diploid cell having this set of chromosome when will undergo reduction division will only produce one type of gamete having autosome plus sex chromosome, that is always going to be Z. So here males are showing homogametic condition. Homo means same, right? But females are showing heterogametic condition because they are producing bachcho two different types of gametes. Clear? What is female heterogamety in case of moths and butterflies? If yes, then let's proceed with the second type of female heterogamety seen in case of birds, that is ZZZW type. The ZZ is representing for males and ZW is representing for females. So one is for male, another is for female. Chalo bhai, dekho kya hoga. So female bird, that is your hen. Hen is going to have a pair of autosome. Along with this, they will have a pair of sex chromosome represented as Z and W. Correct? Now, this is going to undergo reduction division. It is going to show meiosis to produce two types of gametes, one with Z chromosome and another with W chromosome. So here female is showing simply heterogametic condition, but the male birds like cock, it is going to have the same type of sex chromosome along with autosome. When such diploid cell is going to undergo reduction division beta, all the time it is going to produce the same type of gamete having Z as sex chromosome. So here males are showing homogametic condition. Understood? These are the four different methods of sex determination which you see in terms of chromosomal arrangement. Now beta, there are one organisms called honeybee. You all know, you all eat honey at home. Honey wax are available in the market. It's this organism is well known to each and every one of us. Now honeybees beta, they show a very different type of sex determination. So that is why sex determination of honeybee we said study separately. So sex determination of honeybee, first of all, it was studied by scientist Arhino Toki. It was studied by Arhino Toki. Therefore, this type of sex determination is also known as Arhino Toki sex determination. Apart from this, it is also known as beta haplodiplontic sex determination. It is also known as haplodiplontic sex determination. Right? Up, let's see what is special in honeybee. Why it is known as haplodiplontic. Hota kya hai beta? What happens in case of honeybee? Beta, male honeybees are known as drones. They are known as drones. And the fertile female honeybee is known as queen. Female fertile honeybee is known as queen. Beta, drones, they are haploid cells. They are haploid organisms which develop by parthenogenesis. I'll show you how. First, let me write it and then I will explain. Whereas females are diploid, which develop by normal fertilization method. Right? Now, what happens beta in case of honeybee? Check out. Wait, let me add a page first. Okay, check this. Very interesting concept. First of all, a diploid cell of honey is having total 32 chromosomes. right? That means 16 in haploid cells and diploid cells are having total of 32 chromosome. Now, female fertile honeybee, which is known as queen honeybee. This beta, queen honeybee is diploid, having 16 pairs of chromosome, also called as 32 chromosomes, right? Now, this 32 chromosome is going to undergo a cell, a female diploid cell, which is gonad cell, will undergo reduction division, will produce gametes having 16 chromosome, right? Now, what happens? This is about the female. Now, this is the male drone which is going to fertilize with the female honeybee. I told you they are haploid organisms. So, their gametes are going to develop by mitosis. Gametes. Here gamete is going to develop by mitosis. Okay. Now, haploid cell from male honeybee and haploid cell from female honeybee, they are going to fertilize. So, when fertilization takes place, beta, the resultant is always female child. However, if without fertilization, if the female gamete directly develops without fertilization, if one of the female gamete directly develops into a new organism, then such a organism is going to be haploid in nature, and without fertilization, that means they are developing parthenogenetically. So this is parthenogenesis. Right? And they will be called as drones. They will be called as drones. So now can you understand why it is known as haplodiplontic? So in case of honeybee, sex determination is haplodiplontic because males are haploid in nature, whereas females are diploid in nature. Females, they develop by fertilization, whereas males, they develop by parthenogenesis, where female gamete does not undergo fertilization, rather it develops directly. This female gamete, instead of fertilization, will directly undergo mitosis. From one cell it will produce many cells, and those group of many cells will develop into male honeybee, that is drone. So drones are always haploid, but queen bee, female honeybees, they are always diploid. Understood the whole mechanism of fertilization in honeybee. It was explained by Arhinotokey. So it is also known as Arhinotokey type of sex determination. It is unique. Why? Because in the same species, males are haploid, females are diploid. Right? Understood? Chalo. So this much beta you have to do for the sex determination. Now we are going to introduce the next terminology, which is mutation. Beta, the concept of mutation was given by scientist Hugo de Vries while he was working on Oinothera lamarckiana, commonly known as evening primrose. Correct? So the concept of mutation was given by Hugo de Vries while he was working on evening primrose. Scientifically, evening primrose is known as Oinothera lamarckiana. Now, what is this mutation? What is the term mutation means? Beta, mutations are basically sudden change. Sudden change in the genotype and phenotype of an organism. They are discontinuous. Why discontinuous? Because it is not necessary if one thing is present in the parent, then the same changes are going to reflect in his or her next generation. These mutations, they are not properly heritable. They are basically, you can say that mutations can be of two types. Mutation which can be inherited, mutations which cannot be inherited. So usually mutations are not passed from one generation to another, hence they are discontinuous. So sudden means they are not planned. Any change in the environment occurs which can reflect a change in your genotype. And when the genotype changes, automatically the appearance, the morphological appearance, the phenotype of an organism also changes. So mutations are basically sudden, discontinuous changes in the genotype and phenotype of an organism. Now, why genotype is changing? Because maybe there is a change in the arrangement of gene. Maybe there is change in the arrangement of chromosome. Suppose a cell is having normally 46 chromosomes. Now mutation occurred. One chromosome got deleted. Now only 45 chromosomes are there. So 45 chromosome means mutation. Automatically the genotype of an organism will change, and you will also see the change in their phenotype. Understood? So how will you define the term mutation, beta? Mutation is a sudden, discontinuous. It is not necessary that if parent is suffering from a mutant cell, then that mutant cell should pass to the offspring. It is not at all necessary. Discontinuous variations in the genotype and the phenotype of an organism. Why it is happening? Due to the change in the chromosome or the arrangement of genes. Correct? Now beta, mutations also causes variation. Also helps in thus evolution. There are two reasons for evolution. There are two reasons for variations. One, you know, sexual reproduction. Second is mutation. Beta, variation or evolution due to sexual reproduction is very predictable. It is like very slow process. Like in 20 generation, a change will appear. Like initially we were monkeys and then we came to as like these human beings. This variation, this evolution from monkey to Homo sapien is actually due to sexual reproduction. With every generation, some changes were occurring. Those variations, those changes, they got accumulated, keep on accumulating, and all of a sudden, a new organism is produced. So basically, evolution due to sexual reproduction, my dear student, is a very slow event. But mutation can bring evolution in a very minimal time, maybe in one generation only. Suppose my offspring comes out as a mutant, as a complete different organism. So in one generation only you can see a different organism produced from the parents. You must have seen all these movies, all these Spider-Mans and these Batmans, all whatsoever, all your that Green Man, Hulk, all these are mutant people. They are just created due to some mutation occurs in some or the other body cells, and you see how a complete new organism is produced. Now you cannot call Hulk as a normal Homo sapien. Definitely in his body there will be some different number of chromosomes, different arrangement of gene, right? So mutation is just bringing the evolution in a sudden manner. You don't have to wait for long. So there are two ways to produce evolution. One you know is sexual reproduction and second is mutation. Correct? Beta, there are three types of mutation which you have to study. First is gene mutation. Second is chromosomal mutation. And third is genomatic mutation. So how many types of mutation are there? Three types: gene, chromosomal, genomatic. Today I am going to teach you about gene mutation. These two we are going to do in our next class. Gene mutation. Beta, basically, see what happens. This is the chromosome. In a diploid cell, this is a pair of chromosome. Now, these chromosomes are basically having what? They are having genes. Beta, genes are the compacted form of DNA. Genes are nothing but the compacted form of DNA. And what is DNA? DNA is a polynucleotide. It is a basic, DNA is basically a sequence of nucleotides. Right? Now, suppose this is gene 1. Now, you just open this. This is compacted form of gene 1, where you see. Suppose you open this gene 1. Now, this gene 1 will be made up of so many nucleotides. So many means 4 types of nucleotide present in different arrangement. Adenine, thymine, guanine, adenine, thymine, guanine, cytosine, 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 cytosine. So, this is how gene 1 is present. So basically, you know, in our nucleus is very, very small. But the length of the total DNA is very, very long. The length of the total DNA is 2.2 meter. But the nucleus is very, very small. So if you really want to keep that long DNA inside the nucleus, our genetic material has to be in coiled state. So basically, the genetic material in nucleus is this chromatin fiber, which is a highly condensed state having compacted form of DNA called genes. Suppose I open the gene, I will see different nucleotidic arrangement. Now, what happens in gene mutation? Gene mutation means when mutation occur in the gene of a chromosome. And where in gene? When mutation occur in the sequence of the nucleotide of one gene, then it is known as gene mutation. Suppose a normal gene 1 should have this arrangement of nucleotide. Mutation occurred, cytosine is replaced by guanine. Now, one nucleotide only changed, but mutation occurred. You will see lot of difference in the organism. The best example is sickle cell anemia. Right? So, what is gene mutation, beta? When mutation occurs in the nucleotide of a gene. Right? So, when the change occurs in the nucleotide of a gene, then it is known as gene mutation. Correct? Now, gene mutation, beta, can be either point or gross. Point means when only one nucleotide of a gene changes. So, what is point mutation? When one nucleotide of a gene undergoes mutation. Whereas, if number of nucleotides, suppose all these 4 cytosine are replaced by guanine, then mutation is occurring in one gene, but not in one nucleotide, in number of nucleotides, in more than one nucleotide. So, when more than one nucleotide undergoes changes, then it is known as gross mutation. Gross mutation under gene mutation. Clear so far? The best example of point mutation is sickle cell anemia. I will teach you when I'll teach you pedigree analysis what is sickle cell anemia. But for time being, just remember point mutation, sickle cell anemia. Okay, chalo. Now, more divisions of gene mutation, more categories of gene mutation. Frame shift mutation, Frame shift beta mutation, under this type of gene mutation we have two type, that is deletion frame shift mutation or addition frame shift mutation. Deletion means when two or more nucleotides of a gene get deleted. Suppose two cytosines, they got deleted at the end. So obviously there will be a mutation. That mutation will be deletion type of frame shift mutation. When two or more nucleotides gets deleted. Addition means simply when two or more nucleotides gets added. When two or more nucleotides gets added in the gene sequence, bringing mutation. Clear? Yes or no? This is about the two types of gene mutation under the heading frame shift mutation. Now, apart from deletion addition, there is substitution mutation. Substitution means neither the nucleotide number is not going to change, but the quality of the nucleotide is going to change. So in substitution mutation, number of nucleotide in a gene will remain same. However, the type of nucleotidic arrangement will change, will remain same, but the type is going to change. Right? Understanding beta simple English? Now, they can be of two type. These two, substitution type of gene mutation beta can be of two type. Transition type of substitution gene mutation and transversion type of substitution gene mutation. What is beta transition? Transition means, okay, first of all, do you know what is purine and pyrimidine? Let me tell you beta, our DNA, it is made up of 4 nucleotides, which are adenine, guanine, thymine, cytosine, right? Adenine pairs with thymine, guanine pairs with cytosine. Adenine and guanine beta, they are called as purines, and thymine, cytosine beta, they are called as pyrimidines. They are type of heterocyclic organic compound. Purines and pyrimidines are heterocyclic hydrocarbons. Basically, they are organic compounds. Adenine, guanine are purines. Thymine, cytosine are pyrimidine. This you study in detail in class 11th chapter biomolecule, if I am not wrong. Yes or no? Chalo. Transition means beta when a purine is replaced by purine or pyrimidine is replaced by pyrimidine. That means, for example, initially, if adenine was present on the nucleotide of a gene, now it is replaced by guanine. So mutation occurred. Number of nucleotide remained the same. However, the quality changed. Instead of adenine, guanine came up. So purine is replaced by purine. Suppose initially there was thymine. Now cytosine came up. Number remained changed, but quality differed. Now pyrimidine is replaced by pyrimidine. So that is again under transition type of mutation. Correct? Now next is transversion. When purine is replaced by pyrimidine and pyrimidine is replaced by purine. That means if suppose adenine is there and now it is replaced by thymine. Guanine was there, now it is replaced by cytosine. So when a purine due to mutation is is altered by pyrimidine, or pyrimidine due to mutation is altered by purine, then this type of substitution mutation, gene mutation, is known as transversion. Am I clear with all the types of gene mutation? What is gene mutation and its type, point, gross, frame shift, deletion, addition, substitution, transition, transversion? If yes, then I am going to keep my class for today till here only. Again, simple and crisp. The remaining two types of mutation plus the introduction of pedigree I am going to give you in my next upcoming class. So stay tuned with ma'am. Keep learning, keep enjoying the classes. Yes, you know what is the trend. At the end, you do get the homework questions to solve. So make sure that you are solving these questions. If you face any trouble, then let me know. I am going to help you with that. Meanwhile, thank you for joining me. Bye-bye everyone. Take care.